we've become quite interested in the connections between the microbiome and neurological states, and most specifically in animal models of autism or autism spectrum disorder. And the way we got into this area was really almost, sort of a hunch more than anything else, based on the fact that there are many similarities between the immune system and the nervous system. So if you think about all the different organ systems of the body, these are the two that learn from their environment. They have memory, right? And on a molecular and cellular level, there's a lot of overlap between how the two systems work. And so, as you may know, cytokines are widely studied as immune molecules, but they are both sensed and produced by neurons. And conversely, neurotransmitters are, exclusively studied by neuroscientists in the context of neurobiology. But almost all cells have receptors for neurotransmitters, and many cells produce neurotransmitters. And in fact, if you go back about 30 or 40 years, there's literature showing that immune cells actually synapse with cells with neurons. So in other words, immune cells and cells of the nervous system actually touch each other and presumably communicate. It's a very still an understudied field. And so I think those were sort of the initial pieces of evidence that made us think that if microbes were impacting the immune system so profoundly, that perhaps it's not such a huge leap of faith to believe that similar associations were occurring with the nervous system. And over the past several years, over the past, like I said, 5 years, we've been able to show that indeed in mouse models that gut bacteria do affect conditions related to autism spectrum disorder, as well as anxiety, which is related to autism, but also separate from autism, and has really, in many ways, sort of captivated our imagination for how gut microbes can be used to really understand behavior, psychiatric disorders, and even neurodegenerative disorders. We work on Parkinson's disease and Alzheimer's disease as well. 70% of your peripheral neurons, let's say the neurons that leave your central nervous system and go into the periphery, are in your gut, right? So 30% of the rest of your body. So if you think about all your touch and taste receptors and pain receptors and all the neurons that are making your heart beat and you breathe, that's only 30%, right, of your neurons. 70% are in, go to your gut. And so, of course, many of these are involved in gut motility, right? But bugs have access to our brains, right? And through the enteric nervous system, via the vagus nerve, they have direct access to the central nervous system, the brain, and the spinal cord. So microbes are involved in the development of the immune system, which, once again, 10 years ago was heresy to even suggest, because, we all grew up thinking and believing, because it is true, that the immune system evolved to repel microbes, to fight off infectious agents, right? And so it's just sort of this perspective that there's this one-way communication, that any microbe that is sensed by the immune system or recognized by the immune system would immediately be attacked, right? But the work that we did, which I think really opened this field, is show that the dialogue goes both ways, that gut microbes impact the development and the function of the immune system itself, whether it's on a cellular level, right? There are different cell types that develop under the instruction or education of the microbes, functions in those cell types that develop under the instruction of microbes, as well as entire tissues and organs. So if you look in a sterile, germ-free xenic mouse, its tissue architecture is underdeveloped, even in an adult animal, right? And so if you look at the spleen or different immune structures in the gut, they look very immature, even in adult animals. As we understand that, we can think about ways for augmenting immune function, fighting off infections, therapies for autoimmune or inflammatory or allergic disorders, right? And so I think that the role of gut bacteria in improving the development and function of the immune system is something that we can harness for new therapies. So our laboratory has been studying a particular molecule called polysaccharide A, which is produced by Bacteroides fragilis, which is a common human gut bacteria. And this is work, once again, that goes back about 12 years or so now, where we discovered that a particular molecule from this organism, Bacteroides fragilis, was both required and sufficient for the development of the immune system. And when we started this work, it was initially, very much at sort of a very coarse level, because we were just trying to understand various different effects on how PSA, polysaccharide A from Bacteroides fragilis, affected the development of the tissues or of specific tissues in specific anatomical locations. But more recently, what we've discovered is that PSA is very specific in the cell types that it interacts with. And what PSA does is it augments the function of a particular immune cell called a regulatory T cell. And so to take maybe one step back is our immune system can be broadly characterized as having cells in the innate immune system as well as the adaptive immune system. I won't go into the finer details of that. And within the adaptive immune system, there are both B cells and T cells. And these are the cells that that learn from interactions with microbes and have memory. And so in trying to understand the role of PSA in augmenting immune function, we asked initially, is it working through the innate immune system or the adaptive immune system? It's by and large working through the adaptive immune system, though there's an innate component, but that's secondary. And in particular, what we started asking is, are there different subsets of adaptive immune cells that PSA was directly interacting with? And in fact, there's a lot of specificity to PSA's activity. PSA improves the function of a specific category of immune cells called regulatory T cells, right? So within even the T cell population, there are various different subsets, and each of those subsets have different functions. And so by and large, most of our T cells are pro-inflammatory in nature. And so these are sort of the weapons of our immune system. And the reason why they're pro-inflammatory is that they mount an inflammatory response when there's an infection, right? And the immune system is a very, very powerful, has very powerful functions because, if you get a cut on your hand, let's say, you see that there's a tremendous amount of tissue damage during both the control of the infection as well as the wound healing process. You can visibly see host damage. And so as the adaptive immune system is trying to fight off microbes, it's also causing damage to our own cells, right? Because many of let's say, the weapons of the immune system can't fully distinguish between a microbial cell and our own cell, right? And so, in fact, what we now know is that if the immune system becomes activated in the absence of an infection, this is the cause of allergic and autoimmune diseases, right? So the cause of, let's say, asthma or food allergies on the allergic side, or of multiple sclerosis and lupus and type 1 diabetes is an adaptive immune system, primarily the T cells, that have become activated and cause a cascade that leads to tissue damage that results in the symptoms associated with these diseases. And so where PSA comes in is that PSA activates the other arm of the T cell response, the other arm of the adaptive immune system, which is the anti-inflammatory arm. And so the reason why I don't have multiple sclerosis, or the main hypothesis for the reason why healthy people don't have multiple sclerosis, is that we have a good balance between the pro-inflammatory cells that cause tissue damage and then the anti-inflammatory cells, which are the brake on the immune system, which prevent uncontrolled inflammation. And unfortunately, once again, this is still a hypothesis, but there's a lot of evidence for this. Unfortunately, what goes wrong in autoimmunity is that there are defects in the regulatory cells, the brakes, so the cells that keep the immune system in check. And what PSA has evolved to do is to augment that particular cell type, which induces an anti-inflammatory response. And so the two aspects to this that are really exciting, in both in terms of knowledge as well as potentially of therapies, is that what PSA represents is a novel paradigm in biology. Is that there are microbial molecules which activate beneficial arms of the immune system, unlike what's been studied more prominently are microbial molecules which incite inflammation, and these are virulence factors, right? So here's a microbial molecule which actually induces an anti-inflammatory response as opposed to a pro-inflammatory response. And the other aspect, as you might imagine, based on this description of why this is exciting, is that if we can now harness this activity, we can boost the regulatory T cell response. We can boost the anti-inflammatory response, which is missing in patients with allergies and autoimmunity. And we are hopeful that in the next 2 or 3 years, we can start clinical trials using PSA as a therapy for human diseases. Because we've already shown in animals that PSA both prevents and treats inflammatory bowel disease and multiple sclerosis.